My name is Cristina Berchio, and I am a research fellow in the group of Psychiatric Neuroscience at the University of Bari. In this project, we investigate the interaction between biological sex and neural visual processing in different psychiatric disorders. The interaction between biological sex and psychiatric disorder are complex and can explain important clinical variables such as symptom severity, prevalence, age, and onset. However, these aspects are only rarely investigated. In this project, we have decided to study this topic using a specific method called microstate analysis. This is a data-driven method that allows us to explore the neural step of brain process and to reduce complex signal into specific variables. Our group has recently successfully used this method on magnetoencephalography data in a sample of a healthy subject. Visual evoked brain responses were acquired using two different techniques, EEG and MEG. The experimental task used is called the checkerboard task, which involved rapid presentation of black and white stimuli, essentially a checkerboard pattern. This task is capable of evoking very precise visual responses. EEG and MEG data were collected from a large sample of healthy male and female. Additionally, for MEG data were collected from a group of patients with psychosis, while for EEG, data were collected from a group of patients with ADHD, bipolar disorder, and borderline personality disorder. For a healthy subject, clustering analysis showed that can mean effectively separate 4 stages of visual projects in both EEG and MEG signal. This visual processing stage, referred to as microstate, were associated with specific brain networks that were modulated over time. The graph displays the microstate and the corresponding network over time. Different colors represent different microstates, while the associated networks are labeled with numbers. The 4 microstates were also identified in the 4 clinical groups considered. Regarding the comparison between healthy male and female, the 4 microstates were compared in terms of their global explained variant. The data for male are plotted in black, the data for female are plotted in red. This analysis reveals difference between a healthy male and female at the level of microstate 3 and microstate 4. For MEG signal, a bootstrapping procedure showed that healthy male differ from male with psychosis at the level of microstate 4. For EEG signal, this analysis reveals significant difference in the comparison between healthy male and patient. Specifically, significant difference were mainly found for microstate 3. To conclude, our findings demonstrate that both MEG and EEG data identify 4 distinct visual microstates. Alteration in MEG microstate 4 might signal a vulnerability factor in male with psychosis. Changes in EEG microstate 3 could indicate a similar vulnerability feature in male across various psychiatric conditions. This preliminary study highlights the importance of accounting for brain difference between male and female. It also provides further evidence of sex-related difference within functional visual network across psychiatric disorders. I would like to thank those who contributed to the realization of this project and FAIR for the opportunity to implement